Good day, life science students. Welcome to video lecture episode 21. This is going to be the first of the last couple of lectures, and in these lectures I'm going to review the content we had covered since January, just touch on some of the high points and just review any material that you may or may have forgotten, but I think that would be very helpful as you get ready, as you prepare to go to the next grade level, that being 8th grade. Now, remember we started off in chapter 7, 9, and 11. We talked about bacteria and plants and plant processes, okay? Now, remember the big ideas we talked about. We introduced to bacteria, we got an introduction to plants, and then we talked about the plant processes. So let me talk a little bit about bacteria in the general sense. What is bacteria? Well, remember, bacteria is prokaryotic cells with unique structure and function, and they can, remember, be both beneficial, but there can also be ones that are harmful as well. Now, they're single-celled organisms. They don't have a membrane-bound nucleus, okay? And just keep in mind, being a prokaryote, that just simply means before the formation of an actual nucleus, they just have circular or genetic information inside there. Now, we also talked a little bit about, if I remember correctly, we talked about different shapes, right? Remember the shapes we talked about? Well, the basic shapes are, we had coccus, which is round, bacillus, which was rod-shaped, and then spirillum, which was corkscrew-like. Those were 3 types of bacteria shapes we talked about. We also talked about some other structure. We talked about diplo, where there's 2 of them connected together. They're paired together. We have staphylo, which is a bunch together, which is usually because the cell wall is sticky, so they're bunched. And then we had strepto, which is connected to form a chain. Chained. Remember, so then we had coccus, bacillus, spirillum, diplo, staphylo, and strepto. You guys remember that? Hopefully that should sound familiar, right? Alright, let me see what else can I cover in here that I think that would be kind of helpful. We talked about some of the special features, like the capsule that they have, where some bacteria have a thick gelatin, which is an external covering, and it's helpful to protect them from cells that are trying to destroy them and help prevent water loss. The capsule, and then also they have hair-like projections, which could help them stick to surfaces. They have the flagella, which is a whip-like tail to help the bacteria move. Remember that? We talked about that. All right, so, and then we also talked about where they live, that pretty much they can be found in more places than you realize. We also talked about how they produce, that in most case scenarios they go through asexual reproduction, which is similar to mitosis, and then in doing so they produce an exact copy, and that process is called binary fission. Now, there are some that rarely do reproduce sexual reproduction, but most, generally speaking, do it through asexual reproduction. Okay. We also talked about some cyanobacteria and eubacteria. Well, cyanobacteria is a type of eubacteria. Eubacteria are bacteria that are actually helpful. Now, let's talk a little bit, just as a reminder, what cyanobacteria is. Well, cyanobacteria are producers that live in aquatic habitats and are an important food source for the aquatic life that live there. All right, just a quick little reminder there. All right, let me see. Let's talk about aerobic versus anaerobic bacteria. Remember, aerobic bacteria is where they use oxygen while breaking down food, where anaerobic does not need oxygen to break down food. Okay. Remember, there's gut flora. That's the healthy bacteria in our intestines. Remember that even bacteria is used in what we call bioremediation to remove pollutants, like decomposers are used to get rid of oil spills. And it's also in the environment to help maintain balance by decomposing dead tissue, breaking it back down. All right. Just want to remind you of these as well. And then we also, remember, we mentioned about archaeobacteria. Remember, those are the ones that live in those hot springs. They give those unique colors. All right. Then we talked about bad bacteria, right? Remember, those are pathogens. Patho means disease, and gen means to create. So they create disease. So pathogenic bacteria, remember, may harm or kill healthy cells. They may produce toxins that could harm the cell, and they can, of course, be lethal. All right. Now, we talked about some examples. We talked about strep throat, tuberculosis, anthrax, just to name a few. We also talked about how antibiotics are used to attack only bacteria, but you can't use those for viruses. And also the process which we use to actually kill microbes, that is mainly bacteria, is called pasteurization. All right? And I think that should be a good place to stop today as far as reviewing some of the stuff that we've covered earlier in the year. If you have any questions, you can leave it in the comments below. Send me an email. Hope this is helpful. Take care. Goodbye.